BTK degraders is a new class of agents which emerged recently. They are also called PROTACs, proteolysis-assisted targeting chimeras. And a BTK degrader essentially consists of a hook which binds to the kinase of interest, in this case BTK, and a harness which engages the E3 ligase, which is responsible for the degradation of the whole complex. So this molecule linker approximates the BTK, the kinase, and the E3 ligase and shuttles the whole complex to the proteasome, which results not just inhibition of the BTK, but its complete degradation and loss from the cell. So of course, why this is important is because BTK degraders would not necessarily care for BTK mutations. So they will degrade the BTK mutant kinase just as well as they will degrade the BTK wild-type kinase, and preclinical data has shown that. And this is important because we know that mutations in BTK is an emerging mechanism of resistance in patients with CLL who are treated with BTK inhibitors such as ibrutinib, acalabrutinib, zanubrutinib, and even the non-covalent BTK inhibitors such as pirtobrutinib. So this study enrolled several dozen of patients, a phase 1 trial of NX-2127, which is a BTK degrader, which also possesses some immunomodulatory activity due to modulation of the E3 ligase. So the study enrolled several dozen of patients, and including patients with CLL and non-Hodgkin lymphoma. Has shown preliminary efficacy in both CLL and non-Hodgkin lymphoma. So far, there are a few patients with DLBCL have been enrolled, and response assessment thus far is ongoing, but we have a very impressive response in a patient with transformed DLBCL, a patient who was initially diagnosed with lymphoplasmacytic lymphoma, and which then transformed into DLBCL, non-GCB type. This patient achieved a complete response, and after about 6 weeks on therapy, which is currently ongoing. The treatment is also associated with fairly favorable toxicity profile, where main toxicities are very much ibrutinib-like, with predisposition towards bruising, some atrial fibrillation, hypertension, fatigue, etc. So very, very much BTK inhibitor-like toxicities. It's also an oral regimen, which means that it's pretty easy to administer to patients. So in sum, BTK degradation is a very important novel pathway which holds big promise in therapy of CLL and lymphoid malignancies.